Welcome to this explainer. Look, if you follow modern science, you're probably bombarded by the hype surrounding genomics and AI. It's everywhere, right? But today, we're cutting straight through that noise. We're going to look at the actual day-to-day reality of bioinformatics to answer one big question. What does it actually take to code the cell? Because, behind all those massive buzzwords, next-generation sequencing, big data, all of it, there's an actual scientist sitting at their desk. So what exactly does a bioinformatician do in real life? What does their workflow actually look like when they grab their coffee and start their day? Well, to understand the job, we first have to look at the massive problem facing modern laboratories today. Section 1, the biological data deluge. Modern labs are just generating an overwhelming amount of biological data. I mean, the pipeline starts with high-throughput DNA sequencing, then it flows right into complex RNA analysis or transcriptomics and extends all the way through some really intricate protein studies. A single patient sample doesn't just give us a simple yes or no anymore. It generates millions, sometimes even billions, of distinct data points. It is a staggering, relentless output of multi-omics information. And here's the thing, the sheer volume of raw information, it is completely impossible for any human being to process manually. You can't just stare at a spreadsheet of 3 billion DNA base pairs and hope to spot a single pathogenic mutation. It would literally be like trying to find one typo in an entire library full of books where all the books are written in a 4-letter code. It absolutely requires an entirely different computational approach. Which brings us directly to our solution. Section 2. Decoding the chaos. See, there's this massive gap between the messy reality of raw biological data on one side and the clean, actionable insights that doctors and researchers actually need on the other. And that gap is exactly where the bioinformatician lives. They're the bridge. They take a chaotic, noisy starting point, often highly fragmented reads straight from a sequencer, and extract the beautiful, structured biological insights we're looking for. This defines their core task perfectly. Data analysis in this context means using computers and software tools to clean that data, analyze the patterns, and identify important biological insights. It's really about writing robust code that filters out all that sequencing noise and mathematically aligns the reads. This allows us to translate highly complex, hidden biological patterns into readable and, very often, life-saving medical targets. But where is all this happening outside of academic theory? Let's look at Section 3, real-world applications. Companies and research labs across the globe rely on these exact methods every single day for high-stakes science. We're talking about fundamental disease research. We're mapping out cancer genomics to identify driver mutations and track clonal evolution. We're accelerating drug discovery, tailoring highly specific treatments in personalized medicine, and of course, driving rapid vaccine development. Bioinformaticians are quite literally writing the scripts that push modern medicine forward. It's a real game changer. Now, to achieve all this, the scientist must be armed with a very specific modern set of multidisciplinary skills. Section 4, Mastering the Toolkit. The modern bioinformatician sits perfectly at the intersection of 5 distinct domains, balancing them all to tackle complex biology. Think of it like a giant puzzle. First, you need biology to understand the fundamental rules of the cell and genetics. Then, programming to actually write the automated scripts in Python or R. You need rigorous data analysis to mathematically crunch those numbers and validate statistical models. Plus, visualization tools to present incredibly dense, multidimensional findings clearly. And finally, AI and machine learning to do the whole shebang at scale. It's a rare, incredibly powerful split across the board. And speaking of that final piece of the puzzle, AI specifically supercharges this entire workflow. It allows us to predict diseases, discover viable drug targets, and analyze massive genomic datasets at a speed that traditional methods simply cannot match. It's absolutely crucial to realize that AI isn't replacing the bioinformatician. No way. It's the ultimate power tool. It allows them to iterate and discover at an unprecedented pace. As we wrap up this explainer, the core takeaway is pretty clear. We need to learn bioinformatics step by step, using real concepts, tools, and modern AI-driven workflows. Grasping these fundamentals is your very first step to truly understanding how scientists code the cell. It totally demystifies the magic, revealing the rigorous, systematic process that turns deep biological mysteries into solvable computational puzzles. So I'll leave you with this final thought. As AI continues to rapidly revolutionize these workflows, making our tools exponentially faster and sharper than ever before, what massive, undiscovered biological secrets are just waiting for a bioinformatician to decode next? The code of life is incredibly vast, and honestly, we're really just beginning to read it.